Hello, my name is Maxat Kulmanov, and I'm a postdoctoral researcher at King Abdullah University of Science and Technology in Saudi Arabia. Today, I would like to talk about our recent work called DeepPheno, predicting single gene loss of function phenotypes using an ontology-aware hierarchical classifier. So what is a phenotype? A phenotype is an observable characteristic of an organism arising from its genotype and some interaction with an environment. In this work, we are interested in loss of function phenotypes, which arise as a result of a change in a single gene. For example, if a gene gets knocked out, or there's a mutation that results in loss of function of the gene, and we can see some observable change in an organism. Phenotypes are usually described using natural language, and to make them understandable for a computational model, we use ontologies. Ontology is an explicit specification of a conceptualization of a domain. It is used to describe domain knowledge and consists of sets of axioms that are true in that domain. For phenotype, we have several organism-specific ontologies such as human phenotype ontology, mammalian phenotype ontology, which is mainly used for mouse, fly and yeast phenotype ontology. And we also have an integrated multi-species phenotype ontology such as PhenoNet and UPheno. Phenotype ontologies provide structured vocabulary to describe genes or diseases. And we use phenotype classes to annotate genes and diseases. For example, if we take a FOXP2 gene in human, which is associated with a Mendelian disease called speech-language disorder, this gene has annotations with HP classes such as incomprehensible speech, or motor apraxia, abnormality of face, or delayed speech and language, which are phenotypes of the same disease. And this disease also is associated with similar phenotypes. Why do we need these phenotype annotations? These annotations are useful for different computational tasks, such as understanding disease mechanisms. We can predict gene-disease associations based on phenotypic similarity, or we can predict protein functions, or we can use these phenotypes to prioritize disease-causing variants, or we can use it in drug discovery tests, and etc. And the reason we want to predict these annotations, because experimental identification of phenotypic annotations is very slow and expensive, and it is unethical to do experiments on humans. And only few model organisms have phenotype annotations such as mouse or fly and yeast. And this kind of predictive model, which can predict single gene phenotype associations, can be used to also understand complex diseases, which are associated with several genes. Our main feature in our model is gene function. Gene functions are described using an ontology called gene ontology. Gene ontology has three sub-ontologies: molecular function ontology to describe chemical activities of a gene, or cellular components ontology, which describes the locations that a gene can be active, or a biological process ontology, which describes processes where gene can contribute to. Gene ontology is very large and has many classes, and those classes are also not independent, and they stand in different kind of relations. And our main hypothesis is that functionally similar genes should have similar phenotypes. And based on this, we would like to build a model that can predict phenotypes from functions. So our aim is to design a machine learning system that can map gene functions as a set of gene ontology class annotations and additional features such as gene expressions and maybe other genetic features like interactions to predict sets of phenotypic annotations. We aim to encode semantics of functional annotations that are encoded by gene ontology. And we also would like to preserve the hierarchical dependencies of our predict levels. So we want our prediction scores to be consistent with an ontology structure. In other words, if we, for example, predict a specific class, which is a subclass of a subclass, we want, for superclasses, we want to have higher scores than its children. We also want to use ontology structure to reduce the search space, and we want to employ class dependencies to them. And we also need to combine different kind of features, such as functions and gene expressions. Since not many of genes have experimental functions, in our work we also, as a feature, we also use predicted functional annotations. Previously, we have developed a deep learning based hierarchical classifier for function prediction called DeepGo, and its second version, DeepGo Plus, which also uses a deep learning method and predicts function Go annotations from protein sequence and protein-protein interaction network features. Here we build a simpler neural network which uses fully connected layers to represent a sparse representation of gene ontology classes, and it combines it with expression values, and we use a hierarchical classification layer which encodes ontology structure into its computation to predict classes which are consistent with hierarchical space. This hierarchical classification layer was inspired by our previous model, DeepGo, which also used a set of classifiers that are structured in the same way as a gene ontology structure and encoded and predicted labels in a hierarchical space. But the difference is, instead of having a fully connected layers structured as an ontology, we use one big matrix of binary matrix of ontology structure, which encodes subclass relations, and we use a max pooling layer to ensure hierarchical consistency with the layers. We evaluate our method based on fivefold cross-validation on the data from HPO, and we train DeepPheno using different strategies. We first train our model based on experimental functional annotations, and we train both using our hierarchical classification layer and without it. We then also use electronically inferred functional annotations from UniProt and train our model. We also use only DeepGo predicted functions that use protein sequences to predict functions, and then we predict phenotypes. We also add all these features and train one model using all available functional annotations. And finally, we combine functional annotations with gene expression values. Our results show that our hierarchical classification layer improves the prediction performance, and we achieve best prediction results with our final model, where we combine all functional annotations and gene expression values. In addition to this, we also evaluate our model on Kappa second challenge dataset. We train our model on available data from Kappa second challenge, and we evaluate it on the same benchmark and compare with top 10 performing Kappa 2 methods. And our model has 100% coverage for all the proteins in that challenge, and then we also predict, we give better performance. Furthermore, we also evaluate our predictions to show that they are useful, and we test our predictions in a task of prediction gene disease associations. Here, the hypothesis is that genes and diseases that are associated with similar phenotype are associated. And so we compare our predictions with real annotations from human phenotype ontologies. And we also compare several types of DeepPheno. And we also compare with naive predictions. The real HPO annotations give almost perfect performance, because the diseases and genes that we are testing, they're Mendelian diseases, and they have the same phenotype. However, our model, which uses only predicted phenotypes, can also show some predicted performance, and the naive predictions are not useful. We then also evaluate our false positives to test if we can find any false positive predictions that are associated with a directly interacting gene. And we find that around 50% of our false positive predictions are indirectly associated with the genes through one interacting gene. And we test the significance of this result by running random simulations, and we get the value of 0.02. The limitations of our work is that currently our model uses sparse representation for Go classes. So we use a very large vector for gene ontology classes that is binary. And we do not consider all axioms in the ontology. We only use subclass axioms. And we cannot predict more specific classes that have very low amount of annotations in the databases, because our model needs a training data. In future, we would like to incorporate different types of axioms in the ontology, and we hope that this will help us to reduce the search space even more. We would like to use 3D structure features of sequences to predict protein function, and we hope that this might improve protein function prediction performance, which as a result will also improve our performance. And we would like to also encode protein-protein interaction features with a better model. And we're also looking into ways of adding literature features in our method. We also would like to associate phenotypes directly with loss of function variants in the future. So with this, I would like to thank Bioontology Research Group members for their continuous help and feedback on this work, and my supervisor, Professor Robert Hunter. And thank you all for attending my talk. I will be happy to answer any questions you have.